genome evolution, exon duplications, and exon shuffling. Duplications are not limited up to entire genes or the larger fragments of DNA. Duplications can be in the form of exons or in the form of exon shuffling. Proteins are often collection of distinct amino acid domains that are coded by individual exons of a protein or exons of a gene. The separation of exons by introns facilitate the duplication of exons in the single individual genes. Here we can see that what is the concept of exon duplications. In this diagram, we can see that these are the homologous chromosome and exon 1 is here and the exon A is here and the exon B is here. Both exons, they are separated by the intron. These are the homologous chromosome. Due to misalignment between these two homologous chromosomes, we can see that here is the misalignment. This exon A should be here opposite or in parallel to each other, but actually exon A is parallel to or in the alignment to exon B. So as a result of unequal crossover, here we can see that exon B is duplicated in one of the homologous chromosome and it has been deleted in the second homologous chromosome. So in this diagram, we can see that exon B is duplicated. Another concept about the genome evolution can be from exon shuffling. The exons of gene can sometimes be useful units that can be mixed and matched through the exon shuffling to generate new combinations or to attain the new properties of the proteins. Here we can see the structures of the different proteins. This is the protein EGF, another protein is urokinase, another protein is factor 9, and another protein is plasminogen. In case of EGF, we can see here is a protein domain. This protein domain is present also in urokinase. There is another protein domain. But in case of factor 9, we can see this protein domain is double. So here in case of factor 9, there are two similar protein domains. So these similar protein domains are produced from the similar sequences of the exons that can be due to the exon shuffling or it can be due to the exon duplication. Same is case of plasminogen. Here we can see that there are several similar types of the protein domains. There are 5 similar protein domains and these protein domains, they are produced from the similar sequence of the exons and there is a possibility that those exons, they can be duplicated through the process of evolution. So if we conclude that what is exon shuffling or exon duplication, duplications are not limited to the entire genes or to the larger fragments of the DNA. There can be shuffling or duplication of the single exons of the genes.